Hello, I'm Dr. Sandesh, Nanotech.
Good afternoon, everyone. I would like you to welcome to all for the last session of this conference. Dear delegates, could I have your attention for a moment, please? We have made some changes to the program. The presentation of Dr. Anja Jungnickel is will now take place after the poster and oral presentations. Now we are beginning. Now we are beginning with poster and oral presentation, and immediately after that, we'll have our proposed lecture by Dr. Anja Jungnickel. So kindly bear with us for some time, and we'll get back to you with our scheduled program. Now, regarding poster and oral presentation, I would like to again repeat those rules. for poster presentation and oral presentation, all presentation has to be uploaded on WebEx. The link is already been mailed to them. All the participants are expected to upload their presentation on the WebEx link as per the schedule of the presentation. For oral presentation, maximum time given is 8 minutes. A reminder will be given after 6 minutes. And for poster presentation, the time is given as 5 minutes. I think the rules are very clear now. Now we are starting with our oral and poster session. And after that, we'll have lecture from invited talk from Dr. Anja Jungnickel from Analytik, Jena, Germany. Thank you, dear participants. And we have also shifted our session on a new YouTube link, which is already flashing on the whatever YouTube link that was provided to you. Due to some technical reason, we are shifting our entire session on a new YouTube link. The link for new YouTube session is already been shared with you on Telegram group, and it is also been flashed regularly, continuously on the old YouTube link. Okay, kindly bear with us for that, and apologies from our college regarding that. Thank you all the participants. Now, poster and oral presentation will start as per schedule.
Hello, I'm Dr. Sandesh, Nanotek Research Lab, Department of Chemistry, B.K. Birla College, Kalyan, and I'm very happy to present the paper titled Photocatalytic Activity of Zinc Oxide Nanoparticle for Biodegradation. Metal oxide nanoparticles have attained a great importance due to their unique properties. Zinc oxide nanoparticle is known for its advantageous characteristics, a wide direct band of 3.27 electron volt. Zinc oxide is also a non-toxic, biocompatible and simple and inexpensive to synthesize zinc oxide nanoparticle. And it can be used for the various applications like optical, piezoelectronic, cosmetics industry, mechanicals and thermodynamic, electrodynamics and gas sensing. Also, the zinc oxide nanoparticle acts as a good antibacterial agent. Recent study revealed that the plant extract can be used for synthesis of zinc oxide nanoparticle. The present study was undertaken in order to evaluate the efficacy of neem cake extract for the biogenic synthesis of zinc oxide nanoparticle. In this paper, the photocatalytic efficacy of methylene blue in pure zinc oxide nanoparticle was performed in presence of visible light at different irradiating intervals. In the materials and methods, the preparation of aqueous extract, synthesis of zinc oxide nanoparticle, then characterization of morphology, and then photocatalytic activity. In the synthesis, we have taken this cake from the oil companies, and we dissolve this cake in water, boil it and filter it. We'll get this kind of extract, and that extract we have to mix with the zinc acetate solution with a little sodium hydroxide, mix together, stir it well, and we'll get colloidal solutions of zinc oxide, and then we have to dry it. We'll get the zinc oxide nanoparticle, and this powdered zinc oxide nanoparticle was characterized, and then it can be useful for the photocatalytic activity. In the photocatalytic activity, zinc oxide nanoparticles along with the methylene blue dye is kept in this compartment, and then the light of 60 watt bulb is inserted here, and it is fit with the wax, and then the light is coming and interacting with the help of this light, the degradation of methylene dyes takes place. At the interval of time, we have measured how much the dye is degraded and what is the effect of catalyst on degradation. The first analysis in the resultant discussion, we discussed that EV also confirms that the absorption at 355 nanometer is a peak, which shows that there is a zinc oxide nanoparticle. After that, we have measured the particle size of zinc oxide nanoparticle by using the particle size analyzer, and we found that 99.8% zinc oxide nanoparticles having the size is 43 nanometers. So this peak is indicating. Then we have performed the XRD. XRD also shows that the crystallite size, that is by using the Scherer's formula, we have calculated, so found that the 23.21 nanometer is the particle size of zinc oxide using XRD, and it has an hexagonal structure, and this is the result of the photocatalytic activity. As you increase the time, the rate of degradation in the percentage also increases. After 30 minutes, about 27% of the methylene blue is get degraded with using the zinc oxide nanoparticle. And finally, we conclude that zinc oxide nanoparticle can be prepared by eco-friendly and simple efficient technique. The qualitative and quantitative methods for the preparation of zinc oxide nanoparticle was developed by using the neem seed cake extract. It's a waste material thrown by the oil companies. Then after analysis, we have measured the photocatalytic activity. The zinc oxide nanoparticle showed the highest percentage of degradation of methylene blue in presence of light with zinc oxide nanoparticle. Several works on the photosynthesis focus on the removal of organic pollutant from the wastewater, especially referring to dyes which are toxic to microorganism and aquatic animal. Finally, with thanks to our director, Dr. Naresh Chandra, and our principal, Dr. Avinash Patil, for giving the support in the laboratory. These are the references. Thank you very much.
Hello everyone, my name is Neha Sharma, and today the topic of my presentation is study of biosurfactant produced by organism isolated from kiwi and date using petrol as a source of carbon, and its activity was checked against biofilm formation by Candida and E. coli. Next is aim and objective to isolate, screen and characterize biosurfactant producing bacteria from kiwi and date, and its activity was against check biofilm formation by Candida and E. coli. Objective to isolate, screen biosurfactant producing bacteria from kiwi and date, biosurfactant production, characterize of biosurfactant and biofilm production. The next is introduction. The biosurfactant are surface active biomolecule extracellular excretion by microbes, amphiphilic compound, hydrophilic and hydrophobic region. The types of biosurfactant is glycolipids, pterolipids, saphrolipids, corinomycolic. Next is material and method. Media. Screening of kiwi and date using lactic bacteria differential agar, biochemical test, collab test, gram staining, biosurfactant production, MSM medium, biofilm production, catheter, IV set and characterization of biosurfactant. Next is procedure. 10 gram of date and kiwi were dissolved in sterile suspension. A loop full of suspension was isolated on LBD agar and rugosa agar was then incubated at a room temperature for 48 hours. Colony were inoculated on fresh LBD agar incubated room temperature for 48 hours. Culture and biochemical tests were carried out by confirm the organism colony was inoculated in sterile MSM medium with petrol again as a source of carbon. Incubated at a room temperature for 5 to 6 days, drop collab test was performed with engine oil. MSM crude medium was exposed to the purification procedure. The pH was adjusted to normal HCl using NaCl. Incubated overnight in the refrigerator. Extraction was carried out using ethyl acetate and methanol. After extraction, methanol was evaporated and biosurfactant was dissolved in acetone. Organism was characterized at NFV by 60S RNA sequencing. Organism was exposed to UV and inoculated MSM medium for 5 to 6 days at a room temperature, and same purification was carried out. AST was carried out using E. coli, Candida and Ascaris with extracted UV biosurfactant and without UV biosurfactant. Biofilm formation was carried out in catheter and IV set incubated at a room temperature 24 hours. The biofilm was produced by E. coli and Candida in catheter and IV set incubated room temperature for 24 hours. 3 time wash was carrying using sterile saline and simultaneously spread place was performed. Catheter and IV set was cut into small pieces and incubated biosurfactant room temperature for 24 hours. After agitating again, spread place was done. After each was spread place was done, each time growth was observed and counted. First and second was met growth observed. This all indicated biosurfactant produced date and kiwi. Next is result. The result was carried out by isolating kiwi and date sample on lactic bacteria differential agar with one petrol. The similar layer observed during to the growth of bacteria in this media. Next in colony were performed with some biochemical tests. Catalyzed test is positive, cement citrate is positive. The next is antibiotic sensitive tests were determined on nutrient medium. E. coli and Candida is a so to the zone of inhibition. After incubation carry out, spread place was done. Each was difference in counted was noted. The organism was characterized at NFV informed to the Klebsiella pneumonia subspecies. The next is conclusion. The main purpose of carry out this research was isolated the biosurfactant producing bacteria from kiwi and date using as a petrol source of carbon. In its activity was checked again biofilm formation by Candida and E. coli. The growth of bacteria was carried out on lactic bacteria differential agar. The bacteria found to be citrate, urease, catalase, oxidase positive and nitrate tested. The bacteria was identified by 60S RNA sequencing and the bacteria was concluded as Klebsiella pneumonia bacteria. Thank you all for the listening my presentation. Hello everyone, my name is Neha Sharma and today the topic of my presentation is study of biosurfactant produced by organism isolated.
Hello, I am Samiksha Pawar presenting on the topic of treatment of water using organic waste matter of papaya and apple, a research project conducted under the guidance of Dr. Veena Maheshwari along with Jenu Saji, my group partner, and other peers at Bhavans College, Mumbai. In this presentation, we will be looking at the objectives, methodology, results, conclusion, and future prospects related to the research. This research is mainly based on employing the principles of coagulation and absorption for organic treatment of water. Scarcity of pure drinking water, elevated levels of water pollution, and heavy metals in water make it necessary to find an environmentally friendly option for water treatment. The challenge for researchers is to generate a robust, low-cost, and eco-friendly alternative technique to the presently available ones. Here, we have used papaya seeds as a natural coagulant. The high protein content in these seeds acts as cations to attract the negatively charged impurities, settling down the flux of impurities in the bottom and obtaining a relatively clearer water sample. Secondly, we have used apple peels to absorb heavy metal such as chromium from water. Here as well, the cations present on the apple peels attach to the anions such as chromate, and these anions are thus adsorbed on the surface of the adsorbent. Moving further to the methodology, the water samples for treatment using papaya seeds were obtained from two sources, Bhavan's Lake and Versova sewage treatment plant. Both of these samples were tested for BOD, COD, TSS, TDS, turbidity, and microbial load after and before treatment. The papaya was obtained from the local market and the seeds were washed with distilled water and sun-dried before crushing it finely. The papaya seed powder was added to the samples in the doses of 0.5 grams, 0.75 gram, and 1 gram in 1000 ml of water sample and allowed to stand for 60 minutes. Chromium chloride was added to the double distilled water in concentration of 0.5 gram for 500 ml. This solution was used as a control sample for treatment with apple peels. Now for test, we use the sewage sample obtained from the sewage treatment plant. Quantitative tests were performed for the estimation of chromium in the water samples before and after treatment. The apple peels were also cut into small pieces, washed with distilled water and sun-dried before adding it to both the water samples in the adsorbent dosage of 1 gram, 1.5 gram, and 2 gram with the contact time of 60 minutes. Here, we obtained some promising results. As you can see, a clearer water sample was obtained after the treatment with both papaya seeds and apple peels. There was a considerable decrease in the levels of all the parameters for papaya seeds in both the samples. Here, cumulative graphs of both the samples are shown for all the parameters. For apple peels as well, a considerable decline is seen in the levels of chromium in both the samples. Moving to the conclusion, we conclude that the dosage of 0.75 gram for papaya seeds and 2 grams for apple peels show optimum results. Thus, papaya seeds as apple peels can certainly be successfully employed in the treatment of water. Moving to the future prospects, we see that the local production and usage of these fruits make it an easily available option. Also, this technique is a relatively cost-efficient and eco-friendly solution to their chemical alternative. Various industries use apple and papaya as raw material can make use of the organic waste in purification of the effluent, as well as an additional facility can be developed to employ the resulting waste from such industries in the purification of effluents. Further research can be carried out to explore the different aspects of both papaya seeds as natural coagulant and apple peels as natural adsorbent in the treatment of polluted water. Thank you. That's all from my side.
Hello, my name is Rahul Sharma and my project work is about HPLC studies to develop fingerprint and evaluation of antibacterial activity and phytochemical screening of Caryota urines under the guidance of Dr. Pooja Shinde. Aims and objectives. So the aim is to develop HPLC fingerprint to perform antibacterial activity and phytochemical screening of Caryota urine seed extract. Objectives are screening of secondary metabolites in Caryota urine seed extract to check the antibacterial activity against S. paratyphi A, S. epidermidis, E. coli, and to develop HPLC fingerprint of Caryota urines. It is important as it is not yet developed in any ICMR volume. Introduction of Caryota It is commonly known as fishtail palm. The lofty attractive palm with a smooth cylindrical annual trunk is fast growing, flowering and fruiting time throughout the season. It is cultivated as an ornamental tree and for the preparation of toddy, jaggery and fiber. Traditional medicinal uses fruit to treat gastric ulcer, migraine, headaches, snakebite poisoning as well as rheumatic swellings. Materials and methods. The seeds of plant Caryota were collected from Ismail Yusuf College campus. Initially the seeds were washed and dried. Seeds were powdered using grinder. Preparation of extract. 5 gram fine powder of Caryota seeds in 100 ml methanol is sonicated for 30 minutes. The extract were diluted 5 times to achieve the concentration of 1 mg per ml and used for further studies. Phytochemical screening. The phytochemical screening is used to check the secondary metabolites present in the given sample. It can be done by qualitative chemical analysis using specific reagent for specific constituents. The compound that are responsible for therapeutic effects are usually the secondary metabolites. Phytochemical investigation of a plant material support its pharmacological activities and are useful in detection of adulteration. Screening was done using standard method described by Harborn 1998. Phytochemical screening result and discussion. The present work suggests that the seed of Caryota is source of secondary metabolites, that is alkaloids, flavonoids, tannins, sterols, saponins. Table number shows that the absence and presence of secondary metabolites in methanol and path ether extract. All the extract were found to be rich in phytosterols. Methanolic extracts of seeds powder was found to be rich in various secondary metabolites. This study will helpful for developing pharmacological products for this plant. Antibacterial activity, material and method. So the method used was agar well diffusion, medium was MS, diameter of well 8 mm diameter, sample volume 100 microliter. The positive control was chloramphenicol, plant extract 10 mg per ml, incubation time was 24 hours, the measured in millimeter. Result and discussion of antibacterial activity. So there are 4 quadrants in each plate. First quadrant shows positive control, second quadrant shows negative control, third quadrant shows methanol extract and fourth quadrant shows pet ether extract. Each extract was tested against E. coli, S. paratyphiae, Candida albicans and S. epidermidis. Methanol has shown better antibacterial and antifungal activity against selected strains. More activity was observed against gram-negative, that is E. coli bacteria. HPLC fingerprint, material and method. So the first is preparation of standard solution and plant extract through sonicator, keeping concentration 1 mg per ml. The second is sample and standard application by using Chemac ATS4 applicator. Third is development. Normally the development of solvent system like toluene is to chloroform is to ethanol 4:4:1 by twin through chamber. Then drying. After development 5 minutes 120 degrees Celsius in Chemac TLC plate heater. Then derivatization with anisaldehyd sulfuric acid by Chemac plate dipping chamber. Then again drying after derivatization 10 minutes 120 degrees Celsius at Chemac TLC plate heater 3. And last is photo documentation and scanning after derivatization at 366 nanometer on Chemac visualizer and Chemac TLC scanner. So the result and discussion of HPLC fingerprint. The picture shows 3 different fingerprints of Caryota samples which are plated with different concentration. The solvent system of toluene chloroformethanol showed better band separation. The HPLC chromatogram can be best observed under 366 nanometer after derivatization. 8 separate bands were clearly visible in the chromatogram at different retention factor like 0 on 0.1, 0.4, 0.5, 0.6, 0.9. HPLC fingerprint of seeds can be used as diagnostic tool for standardization of crude drug. Further, different compounds can be separated and studied for its phytochemical and biological analysis. Conclusion. From the present review concluded that Caryota is a valuable medicinal plant. It has various primary and secondary metabolites in different parts of plants which incorporated with different antimicrobial property and medicinal property. HPLC analysis revealed a better separation of individual secondary metabolites. The plant can be used to discover bioactive products. And these are the references which are very helpful while working on project. And special thanks to Ankrom Lab Mulund. Thank you.
Hello everyone, my name is Mansi Raut and my research topic is study of antimicrobial activity, antifungal activity and phytochemical analysis of Catharanthus roseus. My aim of this research is to study and check phytochemical analysis, antibacterial and antifungal activities of Catharanthus roseus flower petal extract. Objectives: to study the presence of secondary metabolites, Catharanthus roseus flower extract, to check the efficiency of flower extract against antibacterial activity and antifungal agents. Introduction: Catharanthus roseus is commonly known as bright eyes Cape periwinkle, graveyard plant, old maid, pink periwinkle, rose periwinkle is a species of flowering plant in family Apocynae. Catharanthus roseus is evergreen shrub or herbaceous plant growing in 1 m tall. As an ornamental plant, it is appreciated for its hardiness and dry, nutritionally deficient conditions. This plant is well known for its pharmacological importance, that is, various parts have anti-cancerous, anti-sterility, anti-helminthic, anti-diarrheal, anti-diabetic, etc. activities. Presence of bioactive compounds include tannins, alkaloids, carbohydrates, terpenoids, steroids, and flavonoids. Materials and methods. Flower collection. The flower petals of Catharanthus roseus was collected from Toss Madan Churchgate, Mumbai, Maharashtra. Extraction of flower material. The flower petals were washed and dried in hot air oven, then was grounded in powder using mechanical grinder. Then 1 gram of powder was extracted using 100 ml of methanol, distilled water, pet ether and chloroform by cold extraction method. The flower extract were filtered, then kept in sterile flasks under refrigerated conditions for 3 to 4 days for further use. Phytochemical screening. The extracts were subjected to preliminary phytochemical testing to detect the presence of different chemical groups of compounds. Air-dried and powdered plant materials were screened for the presence of tannins, saponins, flavonoids, alkaloids, phytosteroids, phenols as described. Phytochemical results showed methanol and aqueous extracts were found to be rich in secondary metabolites. Non-polar extracts showed presence of tannins and saponins. Antimicrobial and antifungal activity. In the present study, agar-well diffusion technique was used to for evaluating the antibacterial potential. The extracts of flower petals were prepared in methanol and distilled water. The antimicrobial activity was performed using 2 solvents using sample as powdered form of Catharanthus roseus flower. Flower extract was tested against E. coli, epidermis, candida, paratyphi A. Screening was done in duplicate mean was calculated, distilled water and methanol. Good antibacterial activity was observed against paratyphi A, C. albicans and E. coli. Aqueous and methanolic, both the extracts exhibited very good antibacterial activity against selected strains. Antibacterial activity is attributed various secondary metabolites that are present in the extract. These were the results of zone of inhibition on the plates. Conclusion: very less work was reported on flower C. roseus, so it was an attempt to tap sources of antibacterial phytochemical activities. Polar extracts of flowers Catharanthus roseus was found to be rich in many secondary metabolites, so this may be considered have good pharmacological activities. Promising antibacterial activities was observed in both extracts, so extracts can be source of potential antibacterial and antifungal compounds. These are the references. Thank you.
Hello everyone, my name is Kajal Wagh. My research topic is study of antimicrobial activity, antifungal activity, antioxidant activity and phytochemical screening of Spectroscopy trilobata. Aim and objective. My main aim is to study and check the antimicrobial, antifungal, antioxidant activity and phytochemical analysis of Spectroscopy trilobata flowers extract. Objective: to study the presence of secondary metabolites in Spectroscopy trilobata, to determine the antioxidant property of Spectroscopy trilobata flowers, to check the antimicrobial activity against S. epidermidis, paratyphi A, candida and E. coli. Introduction. Spectroscopy tylobata, commonly known as baby skin creeping oxide Singapore digitaling design Vedelia, is a plant of Astraceae sunflower family. It is widely cultivated as an ornamental ground cover. Spectroscopy tylobata is a herbaceous perennial plant growing up to 45 cm tall. The plant is harvested from the wild for local medicinal use. Materials and method. The flower of Spectroscopy tylobata was collected. Preparation of flower extract. The flower water even and then was grounded in powder using mechanical grinder. The 1 gram of powder was extracted using 100 ml of methanol, chloroform, PET ether and distilled water and then kept on sonication for 15 minutes. The extract were then kept in room temperature for 24 hours. Phytochemical analysis. The phytochemical analysis is done to check the presence of the secondary metabolites with the help of different methods. In the phytochemical analysis, the present work suggests that the flower of Spectroscopy tylobata is the source of secondary metabolites, that is alkaloids, flavonoids, tannins, saponins. The methanolic flower extract of the plants were found to have the resources of phytoactive compounds as compared to others. As we seen, the positive sign indicate that the presence of secondary metabolites and the negative sign indicate that the absence of secondary metabolites. Antimicrobial activity. The antimicrobial assay was determined by agar-Dix diffusion method. The methanol aqueous extract was checked against E. coli, paratyphi A, Candida and E. coli. In the antimicrobial activity, each extract was tested against E. coli, paratyphi A, Candida and S. epidermidis. Result of this study indicated great variation in antimicrobial activity. Methanol has shown better antibacterial and antifungal activity against selected strain. More activity was observed against gram-negative, that is E. coli bacteria. Antibacterial activity can be attributed to phytochemicals that are present in extract, that is flavonoids, sterol, alkaloids, etc. Antioxidant activity. Hydroxyl radical scavenging method. The hydroxyl radical scavenging activity was measured by salicylic acid. The absorbance of mixture was measured at 510 nanometer using UV visible spectrophotometer. Result and discussion. The methanol, chloroform, petroleum ether by hydroxyl radical scavenging activity, which shows the inhibition percentage in methanol, that is 19.30% and 48% for aqueous extract respectively. The extract observed higher potential in hydroxyl radical scavenging assay. Antioxidant activity if all extract was determined by total antioxidant activity by hydroxyl radical scavenging assay. Conclusion. Non-polar extract of flower has found to be rich in various secondary metabolites. Methanolic extract have shown good antibacterial activity against selected strain. The result indicated that flower extract possesses the antioxidant as well as antibacterial activity. Thank you.
My name is Aishwarya Diwale. My research topic is study of HPLC fingerprinting, antimicrobial activity, antioxidant activity, antifungal activity, and phytochemical analysis of spermidictons suavians. The main aim of this research is to develop fingerprint by using HPLC and check antioxidant, antibacterial, antifungal, and phytochemical analysis of spermidictons suavians. The objective of this research is to study the presence of secondary metabolite and to determine the antioxidant antimicrobial presence in the spermidictons suavians extract and to develop HPLC fingerprint of spermidictons suavians stem extract. Spermidictons suavians, commonly known as forest champa, belong from the family Rubiaceae. Flowering time is October to March. Forest champa is a branch shrub growing up to 1 to 2 meter tall. This plant is seen in Western Ghat and Himalayas from Pakistan to Tibet. This plant is used for the treatment of diabetes, diarrhea, and treating ulcer and wounds. Material and method for this research. The stem were collected from the Sanjay Gandhi National Park and the stem were washed and dried in a shade and grounded in using grinder. The fine powder plant material was then used for extraction. Preparation of extract. 5 gram of fine powder sonicated in 100 ml of methanol for 30 minutes. This extract were diluted 5 times to achieve the concentration of 1 mg per ml and used for the further study. Same method is also used for the preparation of chloroform, distilled water, petroleum ether. Phytochemical screening is done to check the presence of secondary metabolite in spermidictons suavians stem extract with the help of different method for different secondary metabolite. As shown in the table, the Plus indicate presence of secondary metabolite and negative indicates the absence of secondary metabolite. The present work suggested that the stem of spermatogenesis suaveolens is a good source of secondary metabolites such as alkaloid, flavonine, tannin, and saponins. Methanol extract of stem powder was found to be rich source in secondary metabolites. Antibacterial activity of spermatogenesis suaveolens. To check the antibacterial activity of spermatogenesis suaveolens, we use the agar-well diffusion method. Each extract was tested against E. coli, paratyphi A, Candida albicans, and S. epidermis. The result of this study indicate great variation in antimicrobial activity. Methanol extract show better antibacterial and antifungal activity against selected strain. HPLC fingerprinting of spermatogenesis suaveolens. First, we prepare the sample by using sonic heater. Then, second, we load the sample on the silica gel pre-coated aluminum sheet by using Camac ATS4 applicator. Third, we develop the solvent system toluene chloroform methanol in 4:4:1 ratio by using twin trough chamber. Fourth, we dry the plate by using Camac plate heater. Fifth, we derivatize the plate by using Camac plate dipping chamber. Sixth, we again dry the plate by using Camac TLC plate heater. And seventh, we scan the plate at 360 nanometer by using Camac visualizer. The solvent system of toluene chloroform methanol show better band separation. The HPLC chromatogram can be best observed under 360 nanometer after derivatization. 8 separate bands were clearly visible in the chromatogram. Antioxidant activity of by DPPH method. SPTC chromatogram was developed for the flavonoid separation using ethyl acetate, ethyl methyl ketone, formic acid, water in a 5:3:1:1 ratio as a solvent system. It was treated with a 0.4 ethanolic solution of DPPH for evaluating the antioxidant activity. The RF value found is a 0.9, 0.56, 0.7, 0.18. It shows that only few compounds in the extract exhibit bioactivity, and HPLC studies enable us to separate and identify the bioactive antioxidant metabolites. Future perspective for this research. Further study may provide other novel or innovative way of developing antimicrobial agents for this plant. Present work will be springboard for further studies on the purification and identification of active compound present in a spermatogenesis suaveolens rubiaceae. Thank you.
Hello to everyone attending this webinar. I am Dhruvi Nirmal doing my Bachelor of Science in Biotechnology at Mithibai College. I will be presenting on the topic Indigenous Water Purification System for Agriculture Purpose, a research conducted by myself and my classmate Shivani Kamath under the guidance of Siddharth Sagar and Head of the Department Dr. Jayaprada Rao Chundri. In a metropolitan city like Mumbai, it is noticed that agriculture carried out parallel to railway tracks utilizes drainage water contaminated with industrial effluents and domestic waste. This results in bioaccumulation of heavy metals in plants and vegetables. On consumption of such vegetables, health problems like balding, digestive problem, weak immunity, etc. are observed at a very young age. Since conventional water purification system are not affordable by farmers, we are trying to create a water filter which is effective, environmental friendly, and economically feasible to farmers. Vegetable samples were collected to detect the presence of heavy metals in them. Spinach samples were collected from a farm irrigated with sewage water from Goregaon, Mumbai. Sewage water samples were also collected from 3 drainage lines, one from Ram Mandir, other from Vilepale, and the third from an industrial district of Malad. Heavy metal presence was detected in spinach leaves using standard basic test available in Chemical Qualitative Analysis book. Synthesis of filter prototype included testing of 7 filter fabrics for their efficacy to filter the laboratory prepared sewage water containing lead. The prototype was made using the combination of cotton, sand, charcoal granules, and cotton cloth. The efficacy of filter prototype was checked using laboratory made sewage water consisting of lead. Lead concentration was detected by the intensity of yellow color obtained on treating the water with HCl and Ki. The color was colorimetrically estimated at 470 nanometer for qualitative and quantitative analysis of lead in the filtrate. The prototype was also tested with sewage water, wherein the concentration of metals is is low. Hence, we used an instrument called ICP-AES for the qualitative and quantitative analysis of heavy metals. ICP-AES is a leading technology for routine analysis of liquid samples for many elements. The spinach leaves showed presence of lead, arsenic, cadmium, mercury, iron, nickel, cobalt, silver and molybdenum. Laboratory prepared sewage water containing lead was filtered through 7 filter fabrics, namely plastic net, polyester spun, polyester satin, polypropylene spun, polypropylene filament, cotton cloth and nylon. The filtrate were treated with HCl and Ki showing yellow coloration. Then the intensity of the yellow color was compared to that of the stock. It was found that polyester satin, polypropylene filament and cotton cloth gave the best results. Out of the 3, cotton cloth was selected for filter component because it's degradable, environmental friendly and available cheap. The filter prototype was prepared using combination of sand, cotton, charcoal granules and cotton cloth. Laboratory prepared sewage water was filtered through the prototype to check its efficacy. As shown on the graph, the lead concentration was 1 mg per ml before filtration, 0.023 mg per ml during the first use of filter and 0.002 mg per ml by the 8th use of the filter. This indicated the increasing efficacy of filter with use. Qualitative analysis of sewage sample by ICP-AES showed the presence of following metals. Out of the 3 samples, sample IM collected from Industrial District of Malad was qualitatively and quantitatively analyzed for the selected metals. This table shows the concentration of selected metals before and after filtration. These metals were selected because of their harmful effect in human body. It was observed that the filter was effective as it reduced the concentration of heavy metals to less than or equal to 0.01 ppm, which is accepted for potable use. It can be concluded that the vegetables are bioaccumulated by heavy metals, which on consumption are harmful to our health. The filter developed showed 100% efficacy and is cost effective. However, a commercial usage and field trials of the same would give a better idea to the efficacy of the model when exposed to external factors like pressure, and a more sophistication is required to make it user friendly. Acknowledgement. We would like to thank Dr. Rajpal Sripath Hande, Principal, SVKM, Medicinal College, for allowing us to use the infrastructure. A heartfelt gratitude to CEF IIT Bombay for processing our samples in ICP-AES instrument. We thank Mr. Piyush Jaishrani, founder of Filter Service, for providing us with the knowledge regarding filter fabrics. We thank Mr. Nilesh Nirmal and Mrs. Hina Nirmal for helping us collect the sewage samples. These are few of the references used during the study. Thank you.
A very good day to one and all. Myself, Dr. Alkama Faqih, from Rizvi College of Arts, Science and Commerce, Department of Botany. The topic of my research presentation is studies on role of trichome density in foliar dust retention. Dust is one of the major problems in Mumbai city. The main causes of dust pollution and increased suspended particulate matter in Mumbai are industrialization, continuous construction, increased vehicular counts, etc. But there is a solution to every problem. Vegetation can reduce all types of pollution. In fact, foliar surfaces can capture and retain dust on them. The morphological characters such as branching pattern, phyllotaxy, trichomes, etc. can trap a lot of atmospheric dust. The current study is an attempt to determine the correlation between trichome density and dust capturing capacities of some selected plant species. For experiments, dust was artificially blown into a previously constructed dust chamber. Dust fall index and dust retention index were calculated using the following formula. Plants with high dust values were studied for their anatomical features. Trichome density was expressed in number of trichomes per unit area of a microscopic vision. These are the pictures of dust chamber created for experiments. The values of dust fall and dust retain were calculated in these two settings respectively. The 12 urban plants selected for this experiments were Nerium odorum, Hyptis suavulens, Malacra capitata, Calotropis gigantea, Ficus hispida, Ziziphus jujuba, Ricinus communis, Ficus bengalensis, Ixora coccinea, Pedilendus tithymeloidus, Bougainvillea spectabilis, and Ficus benjamina. Given table describes their values of trichome density, dust fall, and dust retention index. This bar diagram summarizes the relationship between dust fall index, dust retention index, and trichome density of the selected plant species. The pictures of foliar anatomy of 12 selected plant species were as follows. These pictures were clicked using 3V microvision camera attached to the microscopes. To conclude, the plants having more numbers of trichomes have showed very high dust capturing capacities. Following plants showed the dust capture with respect to their trichome density: Nerium odorum, Hyptis suavulens, Malacra capitata, Calotropis gigantea, Ficus hispida, Ziziphus jujuba, and Ricinus communis. These are some of the references. The authors are grateful to the University Grant Commission, New Delhi, for financial assistance in major research grants. Thank you so much for your patient listening. Have a good day. Stay home, stay safe.
Hello everyone, my name is Kailash Shinde and the title of the paper is Synthesis, Characterization and Biological Evolution of some new quinoline analogues as anti-breast cancer and antibacterial agents. These are the points I am going to discuss today. Let's start with the introduction. Nowadays, breast cancer is one of the most common type of cancer among all types. This is a global breast cancer statistics data in 2018. There are approximately 2.1 million new cases are registered for breast cancer, and out of that the 6,27,000 women are died because of breast cancer, which is 11.6% rate of all cancer deaths. On the similar lines, there are various antibiotics and chemotherapeutics are available in the market, but most of them are microorganism resistance. So, to treat serious infections caused by microorganisms is another major healthcare problem worldwide in 21st century. Therefore, to overcome these problems, there is a substantial need of development of anti-breast cancer and antibacterial drugs in the market. During the literature survey, we found that among various important heterocyclic compounds, the quinoline and its derivative represent one of the most active classes with a wide range of pharmacological properties. Here I have shown some of the examples of quinoline derivatives with a different biological activities. During the literature survey, we also found that the presence of carboxymide moiety at fourth position of quinoline ring enhances its biological activity. Here I shown some example of quinoline 4 carboxymide derivatives with a different biological activities. Most of them act as a different inhibitors. So from this you can get to know that the presence of carboxymide moiety at fourth position of quinoline ring is very important in terms of medicinal chemistry. So based upon all this observation, we plan and synthesize a novel quinoline carboxymide derivatives. The final target compounds 2A to 2J were synthesized from 2-chloroquinoline 4-carboxylic acid as a starting material in good to moderate yields. The first step is a classical Suzuki coupling of starting material with a 3-chlorophenylboronic acid in the presence of a base, sodium carbonate and palladium catalyst to obtain the intermediate one. The carboxylic acid group of intermediate one is further chlorinated to converted and into corresponding carboxylic acid derivative by using a chlorinating agent thionyl chloride, which is then finally coupled with different anilines in the presence of a base sodium hydride at a room temperature to obtain the final target compounds. All the final target compounds and intermediate were characterized and confirmed by various spectroscopy techniques, that is carbon-13 NMR, 1H NMR, FTIR and mass. Here I shown the spectral data of compound 2B for your reference. This is a carbon-13 NMR, 1H NMR, FTIR and mass. We also check the purity of all compounds by HPLC method. Let's talk about the biological activity result. All the final target compounds 2A to 2J were screened for anti-cancer activity against a breast cancer cell line MD-MB221 by MTT-assay method, and the standard cisplatin and doxorubicin acyl were used. IC50 values were measured. These are the IC50 values. From this data, it is observed that all the target compounds were shown better anti-breast cancer activity as compared to standard cisplatin, but not comparable to doxorubicin acyl. Among various synthesized molecules, the compound 2J exhibited better anti-breast cancer activity. This is the antibacterial activity data. All the target compounds were also evaluated for antibacterial activity by agar-well diffusion method against 2 gram positive bacteria and 2 gram negative bacteria, and the standard streptomycin were used. The zone of inhibition value were measured in millimeters. These are the zone of inhibition values. From this data, it is observed that all the compounds shown less antibacterial activity as compared to standard streptomycin. The compound 2C, 2F, 2H, 2I and 2J show moderate antibacterial activity against all tested organisms. So based upon all this observation, it was concluded that a novel series of quinoline analogues were synthesized, characterized and evaluated for anti-breast cancer and antibacterial activities, and a systematic structure-activity relationship were observed with a varied substituent nature and position. The compound 2J exhibited better promising anti-breast cancer activity among various synthesized molecules. The compound 2J show highest antibacterial activity against all tested organisms among all analogues. So we strongly believe that the current results may be useful in the development of potential anti-breast cancer and antibacterial drugs in future by further modification and optimization of reported molecules. These are the references. Thank you.
Screening of 2,4-D induced teratogenic influence on developing chick embryo, presented by Dr. Rupali Gaikwad, Mahin Khan and Dr. Shoaib. Introduction and literature review. Pesticides are the substances which are widely used to control the pest population. 2,4-D is a commonly used herbicide. These are the chemical analogues of auxins, a plant growth hormone that produce uncontrolled and lethal growth in target plant. They causes uncontrolled cell division in vascular tissues and causes plant death. It is a persistent environmental pesticide, and various authors have conducted the research on 2,4-D toxicity. In study, various material and methods are used. Material used include 2,4-dichlorophenolsodium salt and fertile eggs. Method used includes bioassay method, where initially the range finding test was carried out using different concentrations of 2,4-D ranging from 100 to 1 ppm. Then in second test, detection of phenotypic abnormalities was carried out, where the different concentrations of 2,4-D were selected, where two sets were arranged. In set 1, 1 to 10 ppm concentrations were selected, and in set 2, 0.1 to 1 ppm concentrations were selected to check the phenotypic abnormalities. Plan of work includes the preparation of different concentration of 2,4-D, then injecting these different concentrations in a fertile eggs, incubation of the eggs, opening of the eggs after the incubation period, and identification of the fertile eggs for morphological characteristics. Then the data was statistically analyzed and results were discussed. Based upon above studies, findings includes that the development is badly affected by 2,4-D as reflected by aborted and unfertile chick embryos. In case of bioassay, based on number of aborted eggs, LC50 dose lies between 5 to 10 ppm concentration of 2,4-D. Phenotypic abnormalities, stunted growth and abnormalities development were reported above 0.2 ppm concentration of 2,4-D. Absence of eyeballs and limb bursts in developing chip embryos were reported above 7 ppm concentration. Formation of tumor at abdominal region was reported at 0.9 ppm concentration. Increase in concentration leads to the increase in mass reduction and development of abnormalities. Organ development restricted at elevated concentrations. It is also reported that cutaneous melanoma is distinct at cephalic and abdominal region at 0.5 concentration of 2,4-D. Based upon above studies, our final conclusions includes that 2,4-D affects the developmental process of chick embryo and the process of organogenesis, which is regulated by sonic hedgehog. In case of vertebrates, sonic hedgehog is involved in the process of organogenesis. It also includes the formation of digits on limbs and the organization of the brain. In our studies, 2,4-D treated embryos shows whole body mass reduction at elevated concentrations. Also at higher concentration, absence of eye lobes and reduced limb bursts also reported. These all degenerative symptoms clearly indicates that 2,4-D is altering SS gene, causing overexpression of sonic hedgehog protein. As far as concerned with the cutaneous melanoma at cephalic region, it may be the result of development of weaker immune system of the embryo. It means 2,4-D alters the machinery of immune system also.
अभिमन्यू पठाडे
Here I am going to present my paper entitled An Assessment of Marine Environment and its Impact on Coastal Process and Landform. See, in the introductory parts, the subject of oceanography is a recently developed multidisciplinary study of ocean basins, the oceans and its contents. Also deals with the meteorology, which is the study of atmosphere in and outside the ocean space. The marine environment includes the water of seas, estuaries, the seabeds and its subsoils, and also marine wildlife, its sea coastal habitats. The marine environment is a vital resources for life, and the earth marine ecosystem perform a number of key environmental functions. They regulate the climate, prevent erosion, accumulate and distribute solar energy, absorb carbon dioxide, and maintain biological control. Uh, these are the slides showing you that the objectives and hypotheses of the present study, which are designed to address as follows. Another thing is that in the context of the characterized by the biotic and abiotic components, uh, the organism, biotic components and abiotic components. Biotic components also having the organism and their species, predators and pesticides. Abiotic components, the temperature, the concentrate of the nutrients, sunlight, turbulence and salinity and density. So, in the concept of the estuaries are the coastal areas where the salinity is stored of the meat, the fish and water rivers. Coastal processes, there are the various types of marine ecosystems. In it include the salt marshes, estuaries, the ocean floors, the oceans and the intertidal zones, coral reefs, lagoons and mangroves. The following aspects are studied to marine ecosystems. These are the erosional landforms and depositional landforms, sea cliffs, wave cuts, notches, caves and sea stretches, sea ratchets, arches. These are the erosional landform and these are the depositional landform. Erosional coastal landforms, here I am showing that the sea cliff, wave cut, notches and the another thing also sea caves. An active beach, the area of the sediment subject to transport by wind, waves and currents is divided into three regions by the backshores and foreshores and offshores. A beach is characterized by a coastal upland which can be due to the city of cliff and soil embankment. A fossil burn or an engineering structure such as a seawood or a retreatment. So beach sediment may contain shells and recent to sub-recent like fossil skeletal remains from coral reef. Sound, these things also happen in this content. The coral are the skeletons of the many teeny animals living the side by side. Each one of these animals known as the coral polyp, which is the soft present in the body. These are the reasons for coral bleaching. These are the increased global warming, photosynthetically active radiations, ultraviolet light, bacterial infection, changes in the salinity, herbicides, and low tide exposure. Also, coral reefs that the more subject to continue the bleaching threats that locate in the warm shallows and with a low water flow. In the context of conclusion, the Indian coasts have long series of tropical mangrove forests which may act as source and thing given set of environmental conditions. River discharge into situ biological processes of fertilizer input from the adjoining genetic fields, genetic success influences the nutrient budget in the mangrove. And the forest is mangrove forest is mediated predominantly in the microbial rather than the chemical processes. Another thing is that organic matter undergoes variable degrees these influencing the nutrient budget of the coastal system. These are the references which I taken by me for preparing this research paper. So thank you very much.
Hi, this is Hina here. Today I am going to talk about a topic green bean coffee as nutrient source for insecticide degrading bacteria. This project was performed by the students Pooja and Sanchita as part of their TY dissertation. In this project, we have taken green bean coffee as a source of nutrient, as well as we tried to isolate the bacteria, the normal flora of green bean coffee. Now, in this, the main focus of this experiment was to isolate the normal flora from the green bean coffee and use this organism to degrade the insecticide. We have also taken the insecticide like chlorpyrifos. This we obtain from the general shop. This chlorpyrifos is normally used in household to kill the ants, termites as well as bedbugs. In this experiment, we have chosen green bean coffee. Now, what is green bean coffee? Coffee is generally brown roasted and mature, whereas green bean coffee is immature unroasted. This green bean coffee contains lots of normal flora which can be isolated if we provide a proper nutritional source. To isolate the normal flora of green coffee bean, we have taken MSN medium, that is mineral salt medium, which contain all type of carbon, nitrogen as well as energy source to favor the growth of the bacteria. After isolating the organism, now let's talk about procedure. For this experiment, we have taken 10 gram of coffee bean. We have crushed it with the help of mortar and pestle. After crushing, we have inoculated this coffee in 100 ml of sterile MSN medium. Then we incubate it at RT for 7 days. After 7 days of incubation, we have transferred the aliquot to fresh medium. So like this, we have continued 5 steps. That means 5 weeks. 5 times we have prepared sterile medium and we have inoculated a sample from the previous medium. After the 5th week of incubation, the loophole of the MSN medium was dated out on MSN agar. That too incubated at RT for 24 hours. After 24 hours, we observed some colony. These colonies were picked up and further it was used for degradation. Other than that, the colony characteristics were studied as well as gram staining and various confirmatory tests were carried out. Other than that, we have also performed biochemical test of IMVIC, nitrotase tests, various different biochemical tests were performed in order to confirm the organism. And after cultural and biochemical tests, we have concluded the organism could be Enterobacter. Now this is the slide of, you can see the crushed coffee bean was inoculated in MSN medium, and likewise we have performed, like say we have serially transfer or not serially transfer, we have transfer after each incubation to the fresh medium and incubated. These are the colonies on MSN agar. Very pinpoint colonies we obtain. These are the colony characteristic. On gram nature, we have got gram negative dots, single dots. Now on MSN agar, we have performed the MSN agar. We have picked up 3 different colonies, which was very good, well isolated. These 3 different colonies were selected for degradation. In order to check the degradation, as I told you all in earlier, chlorpyrifos was used. Now to prepare the degradation, we have prepared, to carry out the degradation, we have prepared different concentration of chlorpyrifos. You can see in the table 0.2%, 0.3, 0.5, 1% and 2% with the help of diluent. After that, as I told you all, we have chosen 3 different colonies, colony 1, 2 and 3. After preparing the dilution, readings were taken in spectrophotometer at 0 days. Then after 2 days of incubation, readings were taken, 4 days of incubation and 6 days of incubation. So colony 1, this is about colony 2 and colony 3. Now after each incubation, we have also checked the difference in the reading. As an organism grow in presence of a particular substrate, that the turbidity of the organism increases. So increase in the reading indicate the organism is growing well and there is a turbidity in the liquid, which can be easily make out with the help of difference in the reading, and graphs were plotted for colony 1, 2 and 3, and there was a difference in the graph which indicate the degradation process. Now after using these colonies, what we have done, we have sent this organism to the NCL Pune, CSIR NCL Pune for the sequencing. As we confirm Enterobacter, we were more interested in species also. So 16S RNA sequencing was carried out by NCL Pune and it was confirmed to be Enterobacter jordanensis. This green bean coffee was successful in degrading the chlorpyrifos as well as the organism isolated on MSN agar was confirmed to be Enterobacter jordanensis. The main idea of this project was to isolate the organism from the surface of the green bean coffee. Normally, if you want to isolate the organism, the two basic sources like soil and marine we normally use. But in the coffee bean, like say this part of the experiments was little different, as to isolate the normal flora of coffee bean and use this organism to degrade a particular type of insecticide. At last, we can conclude the main purpose of this research was to isolate, as I told you all, from the surface flora of coffee bean and use for the insecticide degradation. After degradation, chlorpyrifos, as I told, we have prepared different concentration. Readings were taken time to time, incubation and difference, and it was also found out that the best result of degradation was obtained in 0.5% of colony 3. Colony 3 was successfully given the best part of the best degradation process. Let's talk about future prospect. After this degradation experiment, we have also tried to find out chlorpyrifos is converted to some end product. Of course, chlorpyrifos must have been converted to some end product by biochemical pathway. So after degradation, we tried to perform the TLC of the tubes, degradation tubes, and find out which compound it is. But the results were not so satisfactory, as we need some higher technique, maybe HPLC or some other method to detect that particular compound. So future prospect, those who wants to carry out this particular project, they can go and find out the end product and learn more about the biochemical pathway. And to carry out and to find out more and more is future applications. These are the reference what we have followed. Thank you.
Yes, on behalf of on behalf of the organizers, I wish to say sorry for the technical snag. We now move to the session which was delayed due to the technical snag, and hence I would now wish to welcome all the participants and Dr. Anja Jungnickel on for her topic on advances in atomic absorption spectrometry. But before that, I wish to thank Analytik Jena for the great cooperation in making this session and participating in the conference. Thank you, Mr. Sanjeev Jain and Dr. Prashant Dutt for the kind cooperation and coordinating for this session. Let me introduce you to Dr. Anja Jungnickel. Dr. Anja studied chemistry in University of Jena in Germany with focus on analytical and environmental chemistry. She is associated with Analytik Jena since 2000. Previously worked as application specialist for US VIS spectrometer at Analytik Jena, Germany. She has worked on various research projects. Presently, Dr. Anja is working as product manager and an expert on atomic absorption spectrometry products. There are wonderful ranges of this sophistication, atomic absorption spectrophotometer, and the technique. There are many applications also, there are many tools, there are many accessories, and I'm sure in this particular lecture, which would be delivered by Dr. Anja Jungnickel, we will get an insight into this particular tool and this particular science of atomic absorption spectrometry. May I welcome Dr. Anja Jungnickel to deliver her talk. Thank you.
Hello and welcome to the presentation about the advances in atomic absorption spectroscopy. My name is Anja Jungnickel. I'm the product manager of AES of Analytik Jena, and I want to shortly introduce myself. I've studied chemistry in Jena, so I'm a chemist. I have some practical work at the University of Amsterdam, and after that I was working for Analytik Jena as an application specialist for UV-Vis spectrometer. I worked for Analytik Jena in the R&D department, and now I'm the product manager and therewith responsible for the AES system of Analytik Jena. The topics I want to talk about today is first I want to give a short introduction in atomic absorption spectroscopy, just some basics, and after that I have some application examples in environment and food. The first example is the mineral contents in drinking water and non-alcoholic beverages. The second application example is a trace analysis by graphite furnace AES. Then I have a topic about toxic traces of mercury and arsenic. And last but not least, I want to give you a short application example about solid AA. It's a direct solid analysis of food samples or plant materials. So let's start with the basics of atomic absorption spectroscopy. There are three different words: atomic, absorption, and spectroscopy. Hello and welcome to the presentation about the advances in atomic absorption spectroscopy. My name is Anja Jungnickel. I'm the product manager of AES of Analytik Jena, and I want to shortly introduce myself. I've studied chemistry in Jena, so I'm a chemist. I have some practical work at the University of Amsterdam, and after that I was working for Analytik Jena as an application specialist for UV-Vis spectrometer. I worked for Analytik Jena in the R&D department, and now I'm the product manager and therewith responsible for the AES system of Analytik Jena. The topics I want to talk about today is first I want to give a short introduction in atomic absorption spectroscopy, just some basics, and after that I have some application examples in environment and food. The first example is the mineral contents in drinking water and non-alcoholic beverages. The second application example is a trace analysis by graphite furnace AES. Then I have a topic about toxic traces of mercury and arsenic. And last but not least, I want to give you a short application example about solid AA. It's a direct solid analysis of food samples or plant materials. So let's start with the basics of atomic absorption spectroscopy. There are three different words: atomic, absorption and spectroscopy. Atomic means that the elements which wants to be analyzed must be atomized. Mainly metals and semi-metals were analyzed by AES. These metals absorb light and come to an excited state of energy. And then you have spectroscopy. That means you need an optical system to detect the absorbed light. An AES system consists of different components. First, there's the atomizer. Samples are submitted to the atomizer in liquid form in order to dry the sample, decompose the compounds and atomize to get free analyte atoms. Then, of course, you need a light source. Light with specific analyte emission profile is used to excitate free analyte atoms by absorbing specific light. The light goes through a spectrometer. The spectrometer disperses the light coming from the atomizer. And then the light goes to the detector. The detector records the light signal and carries the signal into information about the elements containing in the sample. Another important topic in AES is the background correction, as it is dealing with unspecific signals. There are three different atomization techniques available for AES. First one is flame. It's fast, cheap, robust. There you have a sample consumption in a milliliter range and you reach a sensitivity from milligram per liter to percentage range. There are different types of flames available. A common flame is acetylene air, but also acetylene nitrous oxide flame is used, and here you have few interferences. Another technique is the graphite furnace technique. Here you have a very high sensitivity, that means low concentration. You measure in a microgram per liter range. The sample consumption is quite low. You need just 10 to 50 microliter. And there are different tubes used, pin platform and wall platform tubes, and here we can analyze liquids and also solids. The one disadvantage is that you have to deal with more interferences. And another technique is hydride quartz cell technique. So the hydride system, it's very sensitivity for hydride forming elements in the microgram per liter range. And a sample preparation is necessary to form hydride forming elements, to form the hydrides of these elements. And typical elements are arsenic, selenium, antimony, bismuth, tellurium and tin. Another important topic in AES is the background correction. to correct interferences. Interferences can cause by components of the sample matrix. So there are spectral interferences and non-spectral interferences. And there are 3 different background corrections on the market. The first one is deuterium background correction. It can be used for flame and graphite furnace. And here a deuterium lamp is used, included in the device. It is very simple and inexpensive and no loss of sensitivity. The disadvantage is that the deuterium lamp is limited in the wavelength range up to 400 nanometer. And here it's not possible to correct structured background absorption. The Nova 800 is a system of Analytik Jena which uses deuterium background correction. Then there is the Zeeman background correction. This is only available for graphite furnace technique. It's the most effective correction of structured background. And here we have the Zenit devices of Analytik Jena. A unique background correction is a spectral background correction. This is available also for flame and graphite furnace. And this is the most efficient software-based background correction. And this is unique because only the Contra devices of Analytik Jena with its high-resolution continuum source technique can offer this spectral background correction. AS is a technique to analyze metals, semi-metals and even non-metals from a low ppb to percentage concentration range. There are different application fields. Main application fields are food and agriculture, environment, metals and mining, but also AS is also used in chemicals and materials, pharmaceutical industry and energy power plants. And now I want to give you some application examples in the application fields of environment and food. Typical samples for the elemental analysis here are soils, sediments, waste and wastewater, and also sewage sludge for the environmental part. And for food, of course, typical application samples are meat and fish, vegetables, rice and grain, but also milk and powdered milk. And there are the beverages, drinking water, table water, juices, soft drinks, and also alcoholic drinks like beer and wine. The first application example is the determination of mineral contents in non-alcoholic beverages. The challenge was the direct fast analysis of healthy elements like calcium, magnesium, potassium, sodium and iron in typical samples like juice, mineral water, table water and soft drink. The challenge was a wide concentration range from 5 mg per liter like iron in water and 1000 mg per liter like potassium in juice. And the challenge was also to have a pre-diluted samples and not too many dilution steps. And the solution for that is, of course, a simple and fast flame analysis. So this application can be carried out with the Nova 800, the Zenit 700 or the Contra 800. Flame atomization is robust, fast and inexpensive. It covers a concentration range from milligram per liter to percentage. And the sample consumption is in the milliliter range. There are different burner heads available. There is the 50 mm burner head for acetylene air and acetylene nitrous oxide flame. And for higher sensitive measurements, there is a 100 mm burner head only for acetylene air flame. One benefit of the HA atomizer is the burner head rotation. A burner head can be rotated until 90 degrees. This is impacting the working range, so gives a rotation of 45 degrees can be compared with a dilution factor of about 5. And of course, there are important safety issues like a flame sensor, which monitors all flame gases to ensure high safety standards. And the ignition and also the shutdown of the flame can be pre-programmed in the software. An important accessory for this application is the autosampler. Here, an autosampler was used with overrange dilution function. Recommended is also further flame accessories like the scrapper and the switching valve technology SFS 6.0 to improve safety and performance. The Contour 800 has some additional benefits for this application, like the side pixel evaluation, which I want to explain now. With the side pixel evaluation of the Contour 800, it's possible to attenuate strong absorbance signals like you see here in this animation. So low concentrations are typically measured on the peak of the respective resonance line, but for higher concentration you can measure at the edges of the respective line. So with the highest concentration you can even use this point and calibrate also at this edge of the resonance line and can measure this, your sample. The benefits here are an adaptation of the concentration range, flexible signal evaluation, and with this feature it's possible to determine trace and major elements of the sample in just one method. So this will save a lot of time, and this is possible with less dilution effort. Further explanation you will find in the Keynote. Here you see some measurement results of the determination of the mineral contents in non-alcoholic beverages like the mineral water, table water, different juices like apple juice, grape juice, cherry juice, and also cola. And you see a very low concentration, for example, of iron, mainly in the water samples. Of course, iron gives a strange taste in water. On the other side, you see high concentration of potassium in the juices. More than 1000 milligram per liter were in potassium were in apple juice, grape juice, or also in cherry. And all these measurement results were measured with just one method. This was possible with the Contour 800 features of side pixel evaluation and also the automatic dilution function of the autosampler ASFD. So let's summarize the determination of the mineral contents in non-alcoholic beverages and the benefits of the AJ instruments. The NOVA 800, this application can be done with the FAD model. There is the automatic dilution function and also the performance enhancing flame accessories. Here you see the detection limits, and the total measurement time was approximately 90 minutes. Then this application was also carried out with the Zenit 700P with the same accessory options and a comparable detection limits and also a comparable measurement time. And you see here the benefit of the Contour using Contour F800F or D. It gives best detection limits. The detection limits are much lower compared to Nova and Zenit. And you have the side pixel evaluation, and with this feature you can minimize your total measurement time significantly to just 60 minutes. Another application highlight of the Contour 800 is the determination of nonmetals. Like phosphor can detect it at the line of 213.6 nanometer in very low milligram per liter range, as you see here a phosphorus line. But I want to point to the determination of sulfur. Here we had an application examples where we determined sulfur in wine. And this can be done at the molecular line of carbon disulfide at 258 nanometer. The detection limits which was reached for the wine analysis was approximately 4 milligram per liter. And here you see two different wines which were measured, a red wine and also a white wine, and you see the determined concentration of sulfur and sulfur dioxide here, which were very well comparable. We did a QC spike, and here you see a very good recovery rate of approximately 100%. So an additional application feature is the determination of sulfur, for example, in wine analysis. The next application example is the trace analysis by graphite furnace AES. Some typical food samples like rice, grain, vegetables and animal products were analyzed, as well as samples from environment like soil, sediments and sewage sludge. The toxic elements cadmium, lead and thallium were analyzed in traces of microgram per liter concentration. In this application example, toxic traces of the elements cadmium, lead and thallium were determined in different food samples like grain, rice, fish and animal products. Therefore, the sample were digested using the microwave top wave. We rated approximately 0.6 gram of the sample and added some acids. There were two different heating steps necessary, and after the digestion, the digested solution was transferred to a volumetric flask of 50 milliliter. And of course, the measurement is done with a graphite furnace AES because these samples need a reliable background correction. The graphite furnace technique allows the analysis in a concentration range of microgram per liter, so a real trace analysis. A software-driven temperature program is used, and a furnace camera helps to monitor the drying process. Here on the right side you see typical temperature steps. The first step is a drying process. Here on the pictures which were done by the furnace camera, we see the sample droplet which drops in the tube. Then you see the sample, and the sample is dried, the water evaporates, and then you have just some solid samples in the tube left. The next step is the pyrolysis. Sometimes you have two different pyrolysis steps. And then there is the atomization step at a higher temperature, and after that you clean the tube to be prepared for the next measurement. There are different tubes available. We use transiently heated tubes with pin platform or wall platform. A reliable background correction for difficult matrices is the Zinc technology, where the graphite tube is placed in a magnetic field. With this technique, you first generate the analyte line, including the background correction, without the magnetic field. In the second step, you switch on the magnetic field. What happens is, the magnetic field makes the analyte line invisible, but therefore satellite lines appear instead of. And after that you correct both spectra and you get the corrected analyte line. So this is a technique where you get immediately a background corrected analyte line. Chondrite 100 is a class of its own because it's offering a spectral background correction. There are different correction tools available: reference measurement, also automatic baseline correction, it's called ABC, or the CSI tool, it's a correction of spectral interferences. And there are some benefits for these spectral background correction. It's an efficient background correction also for demanding application. Of course, all correction models are pre-installed in the software, and a big advantage is also that it is independent from the applied wavelength range, and these corrections are available for all atomization techniques, for flame, graphite furnace, and also hydride. Another feature of the Contour 800 is the simultaneous multi-line evaluation. So it offers the opportunity for a simultaneous determination of additional elements. This is caused by the high-resolution continuum source technique with the CCD detector. With this technique, you get a small part of the spectra. You see a small part of the spectra. And in this application example, where cadmium was determined, also an alternative atomic line of iron is available. And you can use this line to calibrate the system with a simultaneously two lines, iron and cadmium, and measure and determine cadmium and iron at the same time with just one measurement. It helps, of course, to save time and costs, and you get more information of the samples. Here you see typical signal shapes for zenith and also for contra. Here the cadmium was determined. This is a signal shape for the standard, and here it's a signal shape for a typical sample. It's a reference material DOM 4. It's the name of the material. The red signal is the background signal, so you see the huge background signal, and only the blue one is the analyte signal. And both devices can use a background correction to eliminate the background signal. The Contour 800 gives a 2D, also 3D spectrum, and with the corrected background you get a very nice analyte line. And here you see some measurement results for reference materials like fish protein, dogfish liver and rice flour. There were certified values available for lead and cadmium, which you see here. And the concentration of cadmium for dogfish liver was a little bit higher compared for the concentration of lead in fish protein or dogfish liver or rice flour. Recovery rates are approximately between 95 and 107 percentage, and we got also very good RSD values. For thallium there was no certified value available, therefore we spiked with 5 microgram per liter and got also very good quality rates, approximately 95% up to 97.6%. Also environmental samples were analyzed. Here we used reference materials of soils and sewage sludge. Sample preparation. We digested the samples with mineral acids like aquaregia or hydrofluoric acid, sulfuric acid, and we used, of course, hot plates or also the microwave. For calibration, we used the function of the autosampler using a stock calibration of stock standard from a stock standard. Also, modifier dosing is optimized by the autosampler, and the autosampler has the overrange dilution function to dilute the sample automatically. Measurements were done with the continuum source as a contra technique. And here you see some results. We did the measurement of lead, cadmium and thallium. Here you see the modifier which we used, and the measured concentration and the certified concentration. And you see of the RSD values and also recovery rates that it was done very well. I will shortly summarize this application example of the trace analysis by graphite furnace AES. On one hand, we used our Zenith series. It has one of the best Zeeman background correction thanks to the magnetic field up to 1 Tesla, and we use the autosampler for automatic calibration and sample dilution. And on the other hand, our Contour 800 series was used. This is a high resolution continuum AS technique with simultaneous spectral background correction. Here it is also possible to use the simultaneous multi-line evaluation to analyze multiple elements with one measurement, and we used also the autosampler. Here you see typical detection limits for the 3 elements. You see very low detection limits for cadmium, lead, and you see for thallium that the Contour has a very good detection limits, even lower than the Zenith series. The next application example is the determination of toxic traces of mercury and arsenic. We had also typical food samples like grain, rice, vegetables, fish and animal products. And the concentration range was also in microgram per liter. The cold vapor technology is used for mercury trace analysis. Here, elemental mercury is produced by the addition of a reduction agent. This elemental mercury is transferred into the detection system by a carrier gas. The atomic absorption spectroscopy as well as atomic fluorescence spectroscopy is suitable as detection. Analytik Jena has a dedicated mercury analyzer. It's called Mercodo Plus, and this device offers both detection principles in one system. The integrated flow injection system enables fully automatic operation. Integrated is also a gold collector for enrichment, which gives very sensitive measurements and best detection limits up to 0.1 nanogram per liter. And with both techniques, this device covers a wide dynamic concentration range. Hydride systems are additional accessories for AES systems. We offer different hydride systems. There is the HS50, it's a very simple hydride system. Then HS55, it's a batch system for difficult matrices. And the HS60 is a flow injection system for a fully automated operation. With this hydride system combined with the AES system Contra, Zenith or Nova series, it is possible to analyze mercury and the hydride forming elements. This is arsenic, selenium, antimony, bismuth, tellurium and tin. With this system, gaseous metal hydrides are formed by the addition of a reduction reagent. After that, the gas with the metal hydride is transported via an inert gas to an electrically heated quartz cell. On the picture here of the right side, you see the quartz cell with the electrical heated, which is mounted directly in the sample compartment of the AES system. The atomization temperature is much lower than compared to flame or graphite furnace application. Here just 800 to 1000 degrees Celsius are necessary. Here you see some detection limits for arsenic or mercury. On the right side you have some norms, the US EPA norm and the German norm for drinking water. The German norm for drinking water has a limit value for arsenic from 1 microgram per liter and for mercury even 0.1 microgram per liter. And you see that the detection limits reached by the hydride systems as well as the mercury analyzer are clearly below the limit values of the standard norms. The best detection limit is achieved with the Mercur Plus with atomic fluorescence spectroscopy using a gold trapping. In the application example, the toxic elements arsenic and mercury as well as the element selenium were analyzed in typical food samples like fish and rice, grain, vegetables. For sample preparation, the top wave was used and the digested solution was also transferred into a volumetric flask. Here you see some results for fish protein. We used also certified reference materials, and the recovery rate are between 97.8 and 103%. We got also very good RSD values, and concentration of mercury in fish protein was 0.4 microgram per kilogram. In rice flour we had a certified reference material for arsenic, and here we determined 0.05 milligram per kilogram with good RSD values and recovery rate. For selenium and mercury in rice flour, there was no reference material available, so we spiked with 4 microgram per liter of each element, and the measured values were below the LOD, and for the spike samples we got also very good recovery rates and RSD values. Another application example is the direct analysis of food samples with solid AA. Solid AA is a unique technology for the AS systems of Analytik Jena for Zenit series and Contra 800 series. It's an accessory for direct solid sampling of solids, oils and waxes. Here you see this accessory, it's the SSA 600, an autosampler for solid samples. With this accessory you can analyze the original sample without dilution, error or analytic losses. You get very fast results thanks to the direct analysis. You need approximately 50 microgram of the sample for the element determination, and integrated is a liquid dosing unit in the system. With this liquid dosing unit it is possible to do a liquid standard calibration or to add automatically different reagents. How does it work? The SSA 600 has a robotic arm, and with this robotic arm the sample platform is transported from the sample tray to the microbalance, where the sample is weighted, and after that to the graphite furnace, where the analysis is carried out. The following video shows the autosampler operation. Here you see the platform tubes where you put on the sample. The robotic arm takes the tube and transports it to a microbalance to determine the exact weight. After that, the platform goes into the graphite furnace, and then the oven program is started. There is a drying process. With the integrated USB camera you can look at the sample material, how it behaves during the oven program. After the drying process you have the pyrolysis step. Some organic material will burn. and then you increase the temperature again to atomize the analytes. And then you measure the absorbance directly. You clean the tube with a heating process, cool down, and after that the robotic arm is transported back to the tray. And what you get is an absorbance line or absorbance spectra. Here cadmium was analyzed in this example. Here's an application example for the direct analysis of food samples. The challenge was that only a small sample amount was available, 10 to 15 milligram. Samples were lyophilized yeast and dried lamina. It's an aquatic plant, as you see here in the picture. And the elements which needs to analyze were silver, copper and cobalt. Sample preparation. It's not necessary to do a sample preparation. Sample material was just lyophilized and crushed and used directly. So no sample digestion was necessary. And here you see some results. Silver was determined in yeast. Typical weight of the sample was 0.05 milligram up to 0.2 milligram. And you see the measured concentration, it was 620 milligram per kilogram. And to calculate recovery rate, QC spike was added to calculate a recovery rate, which was determined here with 97.8. In lambda, the elements copper and cobalt and copper were analyzed with the same weight of the sample, and here you see also measured concentration and recovery rates. Now I come already to the last slices, where I want to summarize the advances in atomic absorption spectroscopy. On one hand, there is the unique high-resolution continuum source AES of the Contra 800 series, and on the other hand, there are intelligent accessories for AES. The high-resolution continuum source AES is using only one xenon short arc lamp, which covers the entire elemental range of AES with improved detection limits. Then there is the multi-element analysis by fast sequential up to simultaneous measurement. The high-resolution continuum source AES allows also an extended working range from sub-ppb to percentage range for metals, semi-metals, and even for non-metals like phosphorus, sulfur, fluorine, and so on. Then there is the high-resolution optic with the CCD detector providing detailed information in 3D spectrum display. And we have a simultaneous background correction with ABC and CSI tool available with the high-resolution continuum source technique. Then there are intelligent accessory. I want to point to the unique direct solid analysis where you can analyze solids directly. We have autosampler operation for automatic calibration and sample dilution up to dilution factor 800. And there is full flexibility with the wide range of accessories to minimize the productivity, safety, and ease of use. For more information about our product portfolio of AES, do not hesitate to contact our homepage. Here you have the link for that. Finally, I thank you for your attention. Greetings from Germany and stay care.
Thank you, Madam Dr. Anja. That was a very self-explanatory presentation. We are glad that many participants have given excellent comments and they would be interested in the presentation of yours. So we would share your PPT. Questions would be answered. I think the participants would be given the email ID of Dr. Anja, and so on the Telegram group, of course. So this was a very good session, which, and I'm sure the young researchers would take the initiative and understand this tool and its application. With this, I thank Dr. Anja Jungnickel and Analytik Jena, Germany, for all this wonderful session. Thank you. On this note, I would like to pay tribute to Dr. Vinay Deshmukh, a marine scientist who had contributed in taxonomy. And on behalf of Ismail Yusuf College, Department of Zoology and Zoology Fraternity, I would wish to pay this tribute as we just got the message that Sir is no more. But understanding his contribution to research and marine science, we give due respect to Sir. We may now proceed with the next session. Till then, we may have a look at the campus slides.
Namaskar. On behalf of Ismail Yusuf College of Arts, Science and Commerce, we are delighted to offer a grand welcome for this valedictory ceremony. We hope you have enjoyed all the sessions and this new synergy of academia, research and innovations will bring out a new revolution. As it is rightly said by a famous poet W.B. Yeats, and I quote, "Education is not filling of pot, but the lightning of fire." A very warm good evening to galaxy of intellectuals. Chief guest of today's valedictory function, Professor Dr. B.G. Kulkarni. Guest of honor, Professor Dr. Yuvraj Malghe. Principal of the college, Dr. Swati Vavhal. Fellow participants, professors, students and various committee members of the conference. I am Dr. Vijay Narkhede, working in the capacity of Head, Department of Statistics, Ismail Yusuf College. It is a matter of immense pride to work in the capacity of co-convener for this unique e-conference. On behalf of the college, we are delighted to offer a grand welcome to you all. We have come towards the end of a memorable 2 days international e-conference on emerging methodologies in pharma, environmental and life sciences organized by botany, zoology and biotechnology departments of our college. We are fortunate to have a wide range of around 2,600 participants across the globe. As it is rightly said, make your lives a masterpiece. You only get one canvas. We are blissful to have one such masterpiece for our e-conference, Professor Dr. B.G. Kulkarni, as the honorable chief guest of the valedictory function. Great minds discuss ideas, average minds discuss events, small minds discuss people. I am profusely elated to introduce one of the great minds, Professor Dr. B.G. Kulkarni, as a chief guest for today's valedictory function. Professor Dr. B.G. Kulkarni is a professor of zoology. He has held a prestigious position as Director of Institute of Science, Mumbai during the years 2009 to 2014. He retired as Director of Institute of Science in 2014. He has been a visiting professor at the University of Mauritius in the year 2014-15. He has been principal of PVG's College, Mumbai during 2015 to 2018. His 12 research papers are published in international journals and 52 in national journals. His 23 students have been awarded PhD degree under his able guidance. He has visited many countries like United States, Argentina, Germany, France and Sri Lanka on fellowship for academic work. On behalf of Ismail Yusuf College, sir, I request you to address this unique virtual audience.
I congratulate Dr. Swati Vahal, Principal of IY College, and her team for organizing a conference on important theme. I hope this conference was a grand success. The theme of the conference, emerging trends of methodologies in pharmacy, life sciences and environmental sciences, is very important theme. Nowadays, when we are fighting against COVID-19 and trying to get a medicine to control COVID-19, instrumentation plays an important role in this regard. A drug on COVID-19 will be possible only with the help of analytical instrumentations. And therefore, the theme of this conference is very important. The conference seems to be a truly international conference because the experts which have given talk on various aspects of methodologies are from renowned institutions of foreign countries as well as India. The subjects like nanotechnology, DNA sequencing, genome theory, HPLC, and other instrumentations are included in this conference. All these instruments are very important in biological research, including pharmacy and other biological sciences. Without instrumentation or methodologies of instruments, research in biological sciences is not possible nowadays. If you visit any research organization or university or a college, most of these universities and research organizations have many instruments in their laboratories. However, these instruments are not utilized on a full scale. I hope the deliberations which has been given by the experts during this conference will definitely inspire the participants to use these instruments on a full scale. The outcome of any conference depends on how the deliberations or views expressed in this conference are utilized in practice. Therefore, the success of this conference depends on how participants will utilize these methodologies in their research. I therefore request Dr. Swati Vahal, principal of this college, to organize such a conference on the same theme every year, or at least after 2 years, to take up a review on this subject of methodologies. Before conclusion, I congratulate Dr. Swati Vahal and her team, including Dr. Vijay Narkhede as co-convener, Dr. Amit Saraf and Dr. Bhavita Chavan as coordinator of this conference, who have taken a lot of efforts to make this conference a grand success. I wish all the best to the Ismail Yusuf College and the team of the conference as well as participants. I thank principal of this college, Dr. Swati Vahal, and the organizer team for giving me an opportunity to be a part of valedictory function. Thank you.
Sir, I am grateful to you for your enlightening words of immense knowledge and experience. Thank you, sir. As it is rigidly said by John Wooden, and I quote, "Things work out for those who make the best of how things work out." We are happy to have an eminent personality, Professor Dr. Yuvraj Malghe, guest of honor for today's valedictory function. It is my privilege to introduce one of the best minds, Professor Dr. Yuvraj Malghe. Professor Dr. Yuvraj Malghe is Professor and Head of Chemistry Department at Institute of Science, Mumbai. At present, he is holding an additional charge as Joint Director, Higher Education, Mumbai Region, Mumbai. He is having a teaching and research experience of 24 years. Dr. Malghe has worked as chairperson as well as member on various committees and bodies like Board of Studies, etc. Sir has organized 8 conferences in the capacity of convener, organizing secretary and coordinator. He has completed 6 major and minor research projects. His 36 research papers are published in reputed journals. 2 students have been awarded PhD degree under his able guidance, and 6 students have registered. He has visited United States, Sweden, Finland, Mexico, and Bahamas on fellowship for academic work. May I request Professor Dr. Malghe to address the unique virtual audience?
Dr. Swati Wawal, Principal of Ismail Yusuf College and Convener of the C Conference. Dr. Vijay Narkhede, Head Department of Statistics and Co-Convener of this conference. Dr. Amit Saraf and Dr. Bhavita Chavan, Coordinators. Dr. Arti Nagarsekar and Dr. Archana Rangari, Organizing Secretaries of the conference, staff of the college and participants of the International E-Conference. First, I congratulate Ismail Yusuf College for organizing International E-Conference on the topic Emerging Methodologies in Pharma, Environmental and Life Sciences. Topic chosen for E-Conference is very good. Pharma industries are important because these industries are major source of medical innovation, and it is also heavily regulated industry. Drugs are evaluated for safety, efficacy and quality. Primary function of the pharma industry is to discover medicines, vaccines and provide it to the society. These medicines and drugs work faster and cure the illness of the patient. At present, entire world is facing difficulties due to COVID-19 pandemic. Pharma industries and researchers are trying to develop medicine as well as vaccine for this disease. Environmental science is important to us because we live in the environment and share resources with other species. Environmental science helps us on how to conserve our environment and face increasing population growth and anthropogenic activities that degrade natural resources and ecosystem. Environmental science is a holistic and multidisciplinary field that integrates the biological, physical and earth sciences. Its goal is to understand how earth works and how it supports life. Life science is the branch of science which involves the scientific study of life and organisms such as microorganisms, plants and animals including human beings. The science is one of the two major branches of natural sciences. Other branch of natural science is physical science. This conference covered emerging trends in all these three important branches of science. I am happy to learn that 2,560 participants are registered for this conference. Once again, I congratulate Dr. Swati Wawal and her entire team for successfully organizing this conference. I am thankful to organizing committee of this conference for giving me an opportunity to interact with the participant. I wish all the best and great success to all of you. Thank you.
I am immensely happy to express my gratitude towards our guest of honor, Professor Dr. Malgesha, for his valuable words of guidance and encouragement. Thank you, sir. As it is rigidly said by John Quincy Adams, and I quote, "If your actions inspire others to dream more, learn more, do more, and become more, you are a leader." The inspiration for this unique way of experiencing knowledge was conceptualized and executed by a person who is not only a great motivation to all of us, but a visionary and the person who gave us the belief to organize this e-conference. Her able guidance brings out the best in us. She is none other than our beloved Principal Dr. Swati Wawal. May I request Honorable Principal Dr. Swati Wawal Madam to address the unique virtual audience for this valediction.
Namaskar and good evening to all of you. I welcome to the valedictory function of the International E-Conference on Emerging Methodologies in Pharma, Environmental and Life Sciences, the chief guest for the session, Dr. B.G. Kulkarni, former director at the Institute of Science, Mumbai, and Dr. Yuvraj Malge, joint director of Higher Education of Mumbai region. I thank you both for accepting our invitation and your words of guidance and appreciation. I also welcome all the resource persons, the participants, and the faculty at Ismail Yusuf College for this function. At the outset, I sincerely thank all our patrons for their timely and constant support and encouragement given to us. I thank Honorable Shri Udayji Samant, Minister of Higher and Technical Education, Maharashtra State, Honorable Shri Prajaktaji Tanpure, State Minister of Higher and Technical Education, Maharashtra State, respected Shri Saurabh Vijay, Secretary of Higher and Technical Education Department, and respected Dr. Dhanraj Mani, Director of Higher Education for the State of Maharashtra. We at Ismail Yusuf College are well equipped with the best of instruments with high precision, which are purchased from companies of international repute. We have LCMS from Waters Company, USA, GCMS of Shimadzu, AAS of Analytik Jena, Germany, the HPLC system from Chemag, Switzerland, the RT-PCR of High Media, the microwave parallel synthesizer from Milestone, and these are just a few being mentioned. I feel that we are privileged to conduct such a conference because we are well equipped with such good instrumentation facilities. These instruments are used routinely by our undergraduate and postgraduate students while doing their research projects. We have good association with industries and research laboratories, and therefore it was possible to arrange lecture of these resource persons of international repute. I take this opportunity to sincerely thank all our resource persons who have shared their knowledge, expertise, and research work with all the participants of this conference. By conducting this conference, the facilities which are available to our undergraduate and postgraduate students were shared with the society. With the aim in mind that the facilities which are available to our undergraduate and postgraduate students, the same should be made aware of to the research students and scholars across the nation, this e-conference was conducted. At the same time, we would appreciate further collaborations for the upliftment of research endeavors. I hope the conference has truly motivated all the participants and has been helpful in their path of research. We are in the process of getting NABL certification for our HPLC laboratory very soon. I congratulate my entire team at Ismail Yusuf College for successfully organizing this e-conference, that too at such a short notice and during the ongoing stressful situation of the COVID-19 pandemic. It is the first time that we have conducted a conference via electronic media. However, certain technical faults did arise, but we should look at our mistakes as lessons and opportunities to do better in the future. I apologize for any delays and technical lapses during any point of the sessions. A formal vote of thanks will be delivered in the end of the conference, but I would like to mention that I am extremely pleased by the efforts which have been taken by all my colleagues. I thank Dr. Vijay Narkhade, Head of Statistics Department and the co-convener of this conference, for conducting timely meetings and encouraging the staff to take up this task. I thank the coordinators of this conference, Dr. Bhavita Chauhan and Dr. Amit Saraf, and the organizing secretaries, Dr. Aarti Nagarsekar and Dr. Archana Rangari, for their effective overall management. A great responsibility was shouldered by the technical committee. I thank Shri Amol Sonawani and his team for taking great efforts and handling the technical aspects of this e-conference successfully. I also acknowledge the efforts taken by the editorial team led by Srimati Swati Phadke for the souvenir and the proceedings of this conference. I am overwhelmed by the large number of participants for poster and oral presentations which were depicting relevant research work. I congratulate all the participants, wishing you further success on the path of research. Very soon, we will publish the full-length papers submitted by the participants in the proceedings of the conference with an ISBN number. I feel that by way of conducting this conference, we have tried to meet the societal needs at its best. I hope that we can conduct many more such events in the interest of the masses. Thank you.
I am extremely grateful, Honorable Principal Dr. Swati Vahad, Madam, for your valuable guidance and encouragement. Thank you, Madam. Gratitude is the fairest blossom which springs from the soul. Every celebration is an experience. The external fades away and the internal lingers and echoes in the deepest part of our hearts. The end of a story is the new beginning for many others. On this note, I take this opportunity to thank all the dignitaries for enlightening us with their knowledge and experience. I shower my heartfelt gratitude to our chief patrons, Honorable Sri Udayji Samant, Minister, Higher and Technical Education, Maharashtra State. Honorable Sri Prajaktaji Tanpuri, State Minister, Higher and Technical Education, Maharashtra State. Honorable Sri Saurabh Vijay, Secretary, Higher and Technical Education, Maharashtra State. Our patron, Honorable Dr. Dhanraj Mane, Director, Higher Education, Maharashtra State. Our beloved Principal, Dr. Swati Vavan Madam, for the encouragement and support. I would love to extend my warm wishes to coordinators Dr. Amit Saraf, Dr. Bhavita Chauhan Madam, organizing secretaries Dr. Nagarsekar Madam, Dr. Archana Rangari Madam, organizing committee members, editorial committee members, advisory committee members, technical committee members, teaching and non-teaching staff who have toiled hard to make this e-conference a great success. Thank you. Jai Hind, Jai Maharashtra.
Thank you, Dr. Narkade, sir. Dear delegates, I have three important announcements to be made. First, regarding feedback forms. A link for the feedback form for day 2 will be shared on the Telegram group as informed. And now for the most awaited announcement regarding the winners of poster and oral presentations. We have accepted 84 abstracts for the presentations under different categories. To start with the winning list, for poster presentation under undergraduate categories, third prize goes to Imtiaz Ahmed Siddiqui. Second prize goes to Sneha Sharma. And first prize goes to Shamima Nazreen Khan. I congratulate the winners. Now, poster presentation for under PG category. Third prize goes to Mridula Nair. Second prize goes to Diana. And first prize goes to Isha Shinde. Again, congratulations to the winners. And third category of poster presentation, third prize goes to Dr. Popat Pathare. Second prize is shared by Kylie Mayers and Vishal Bambole. Whereas first prize goes to Dr. Ranjana Khade. Now, we have received a good response for this. So consolation prize is also kept in this category and it goes to Dr. Susan George. Again, congratulations to the winner. For oral presentations, we have received result only for UG and PG category. The third prize goes to Aishwarya Diwale. Second prize goes to Manasvi Dekate. And first prize goes to Samiksha Pawar. I once again congratulate the winners. Now, the prize for oral presentation in research scholar and faculty category is yet to be announced. It will be announced and it will put up on Telegram group. Now, third important announcement is about the certificate of participation and presentation. Here, I would like to clarify that all those participants who have filled the feedback form and attended the conference shall receive the certificate within 15 days. I would like to conclude that this year's conference was a great success for us because this year it was left with what obvious few thought which we have really learned from the this overall event. First thing which was loud and clear for us that we have to embrace this digital age. We are getting used to webinars, but for many of us, it may be the first e-conference, but I believe it is the way forward. I also thank those who drew up courage and share their research on the online platform. Wishing you all the best. And that's it. Signing off from the Ismail Yusuf College, Mumbai. Stay home, stay safe. Thank you again.